The overall goal of this procedure is to study the effect of mechanical load on the proteolytic cleavage of single proteins in a highly parallel and cost-effective manner. In this demonstration, the cleavage of collagen by matrix metalloproteinase 1 is examined. In the first step, collagen trimers are attached to the glass cover slip of a flow cell, followed by the attachment of micrometer-sized magnetic beads to the individual collagen molecules. The second step is to mount the flow cell into the magnetic tweezers apparatus and check that non-specifically attached beads have been removed from the cover slip surface. Next, the protease is added to flow cells. The final step is to determine the proteolysis rate by monitoring bead detachment in real time. By measuring bead detachment rates at varying protease concentrations and forces, it is possible to derive standard enzymological kinetic parameters using minute amounts of substrate and protease. In addition, this technique provides unique information about the effect of mechanical force on the rate of proteolysis and on the structural changes in the substrate protein that accompany cleavage.
The main advantage of this technique compared to other methods like optical tweezers and atomic force microscopy is that this technique is high throughput and cost-effective.
Though this method can provide insight into the effect of force on the proteolytic cleavage of single proteins, the instrumentation and techniques can be applied to a broad range of biophysical problems, including the effect of force on protein folding and conformation.
Begin flow cell preparation by cleaning cover slips using sonication. Add the cover slips to a small glass container capable of holding them that fits in the sonicator. Fill the container with isopropanol and sonicate in a bath sonicator for 20 minutes. Discard the isopropanol and rinse the cover slips with copious quantities of deionized water.